far, get comfortable. Like you're not gonna be able to hold a squat for very long or rest your arms on something. Okay. All right, so what we have here is a cell. In order to be a cell, you need 3 components, and those are the cell membrane, the cytoplasm, which is the space between the cell membrane and the nucleus, and then the last thing you need is a nucleus. The cell membrane is made up of 3 molecular components. It's mostly lipids. Of the lipids, we have phospholipids, which make up the majority of them, cholesterol, and then glycolipids. Proteins determine the function of a cell. They're found in the cell membrane here. You have 2 types, integral proteins and peripheral proteins. And the last molecular component of the cell membrane is called the glycocalyx. It's an external carbohydrate coat of the cell membrane. All of that you'll learn more about in lecture, but I wanted you to have it on video. The cytoplasm itself has 3 parts. It has the cytosol, which is the fluid, the organelles, and then a large collection of any sort of material known as an inclusion. Looking at the organelles, they're divided into 2 groups, the membrane-bound organelles and the non-membrane-bound organelles. This first tunnel-like network here is known as the smooth ER, and it is a membrane-bound organelle. It produces lipids and detoxifies the cell of alcohol and drugs. The next tunnel membrane here is the rough endoplasmic reticulum, and it's rough because it has these little ribosomes on it, these little red dots, or just dots for some of you. This is in charge of taking the amino acid chain created by the ribosomes and modifying it further to build protein. The rough endoplasmic reticulum will send the protein via a transport vesicle to the Golgi apparatus, which will finish the protein off and then secrete it out via a secretory vesicle. The Golgi apparatus is a membrane-bound structure, and we have these stacked membranes on top of each other that are known as cisternae. Here is a lysosome, which can be created by the Golgi apparatus. It is also a membrane-bound organelle that is in charge of digesting material brought into the cell. Next to it, the smaller or the more petite spherical organelle that is membrane-bound is the peroxisome. It takes hydrogen peroxide created by the cell as a waste product and converts it to water. So the larger one is lysosome, the petite one is the peroxisome. The last membrane-bound organelle in the cytoplasm is the mitochondrion, and it produces ATP. Looking at our non-membrane-bound organelles, all these little dots here that for some reason are in S's are your free ribosomes. They create protein that stays in the cell, while the fixed ribosomes that are attached to the rough ER create proteins that leave the cell, things like insulin. Okay, so, the last one here, we have these bundles of microtubules creating centrioles, one here, or together a centrosome. It moves DNA during mitosis. All right, so those are the organelles of the cytoplasm. The last organelle left is the nucleus. It has 2 membranes, creating the nuclear envelope, and then nuclear pores. It contains DNA on the inside, and then these are nucleoli, which is plural for a nucleolus. These organelles are responsible for creating the ribosomes. The end. I got some more, don't go anywhere. Hold on. Cell membrane, cytoplasm, nucleus, smooth endoplasmic reticulum, rough endoplasmic reticulum with fixed ribosomes, Golgi apparatus, mitochondria, plural, centrosome, nucleus, nuclear envelope, nuclear pore, nucleolus, DNA. The end for this one. I won't ask you lysosome or peroxisome on here because it's hard to see the difference. Cell membrane, cytoplasm, nucleus. Rough endoplasmic reticulum with fixed ribosomes, free ribosomes. Golgi apparatus, mitochondrion, centrosome, nucleolus with DNA around it, nuclear envelope with nuclear pores. The end. One more. Right. Each body cell, each somatic cell, goes through a life cycle that consists of two phases, interphase and mitosis. Interphase is where the cell carries out its normal day activity, protein synthesis, ATP production, DNA replication, so on and so forth. So what we have here is a cell in interphase. The big orange ball that you see is the nucleus. And then that green, or those strings inside of it, are the DNA. Is the DNA. Ooh. It's currently uncoiled because we need to be able to read the genes. Soon after the DNA is replicated during interphase, we'll enter mitosis. The first phase of mitosis is prophase. Notice that my DNA is much more condensed. It's thicker here than it is in interphase. And my centrioles are moving to opposite sides. So this is prophase. Soon the nuclear envelope will completely disappear. The chromosomes will line up in the middle, and this is an indication that we are in metaphase. The centrioles will pull the chromosomes apart, so it'll separate those replicated strands during anaphase. And then the last phase of mitosis, telophase, my cytoplasm starts to pinch off during cytokinesis and a cleavage furrow forms. The nucleus starts to reappear, and my DNA has uncoiled again because we need access to the genes because we're about to enter interphase again and continue cellular work. Protein synthesis, DNA replication, so on and so forth. The end, the end.